and thank you for joining us for our live MDA Engage Duchenne Muscular Dystrophy Symposium. I'm Nicole Petrowski, MDA's Community Education Specialist, and we appreciate you joining us this morning. We're glad to be able to share this important symposium with you. I also want to thank all of our supporters, Avidity Biosciences, PTC Therapeutics, Sarepta Therapeutics, and Solid Biosciences for all their support. It's greatly appreciated. And we believe in the power of bringing our community together for opportunities to learn from specialists and having opportunities to connect with others. And this symposium is part of our larger Engage series with disease-specific symposia that we are hosting today, community education webinars, and seminars. And so here's a schedule of our upcoming community education seminars and webinars that we have for the remainder of the year. The seminars are day-long educational seminars providing individuals and families with knowledge and resources around neuromuscular care. And we have different folks that are going to be experts in the field that will be talking on related topics. And the webinars you see are an hour long, so they're much shorter and they are just focused on that topic at hand that you see. And this is the agenda I have for us today. As you can see, we do have a pretty long day, but we do have a lot of wonderful speakers that are going to be with us. I now would like to introduce to you our first presenter, and I'm sure many of you know who he is, but this is Dr. Craig McDonald. He is a pediatric physical medicine and rehabilitation physician who is also board certified in neuromuscular medicine and pediatric rehabilitation medicine. He is an internationally recognized expert in clinical management, rehabilitation, and precision therapeutics for children and adults with neuromuscular diseases. Dr. McDonald has been a pioneer in the development of novel outcome measures for clinical trials focused on disabled populations. He is widely known for his expertise in the treatment and evaluation of children and young adults with Duchenne Muscular Dystrophy and other neuromuscular diseases. So, Dr. McDonald, I will turn it over to you. Thank you for being with us today.
Great. Thank you very much, Nicole. And I really would like to thank MDA for their support of our care centers over the years. I think we have had support of our care center since, I believe, 1968 at
Wow.
UC Davis. And I also just want to thank MDA for the invitation to speak this morning at this MDA Engage Symposium focused on Duchenne Muscular Dystrophy. Well, today I'm going to focus a bit on potential biomarkers, and I'll discuss what biomarkers are. And the majority of my talk is going to focus on new therapies for Duchenne Muscular Dystrophy. And just as a matter of disclosure, I've done consulting work for Duchenne Muscular Dystrophy clinical trials for a variety of pharmaceutical companies in the Duchenne space. So as you all know, Duchenne is an X-linked condition. It's considered a rare disease. It is relentlessly progressive, ultimately fatal in young adults. And it results in progressive paralysis, ultimately respiratory failure, progressive cardiomyopathy, and premature death. The dystrophin protein is present just underneath the covering of the muscle tissue, the sarcolemmal membrane, and dystrophin really acts as a bridging protein, a shock absorber, which protects the muscle from damage during contraction and stabilizes a number of critical dystrophin-associated proteins. So in Duchenne Muscular Dystrophy, the dystrophin protein, due to the gene mutation, is completely absent. So there's essentially complete absence of the functional dystrophin protein. As a result, the muscle fibers become damaged. Ultimately, the regenerative capability of the muscle fiber is impaired, so the fibers cannot be repaired or replaced, and eventually the fibers are taken over by fat tissue and scar tissue. So as a consequence of absence of dystrophin, there's this mechanically weakened plasma membrane, which is prone to focal tears. during muscle contraction. And there's a massive influx of calcium from outside of the muscle fiber, outside of the muscle cell, and calcium leaks into the inside of the muscle cell, and there's then activation of proteolytic enzymes, which cause degradation, and eventually there's what is referred to as a segmental necrosis that occurs in the muscle cell, where the muscle cell has this focal area of injury, which causes degeneration of the muscle fiber. So in terms of the pathophysiology, there's again a gene abnormality that primarily causes this disease, absence of the dystrophin protein. The muscle membrane is susceptible then to injury, and this leads to a cascade of events leading to muscle fiber injury and degradation. The muscle fiber undergoes cycles of degeneration, regeneration, degeneration, regeneration. And in order for the muscle fibers to regenerate, you have to have activation, or what are referred to as satellite cells, which lie just on the outside of the muscle membrane. And ultimately, the muscle fiber or the muscle cell will die and is replaced by fat and connective tissue. Again, in terms of this, we have the structural defect in dystrophin. The membrane is unstable, the fiber undergoes necrosis, and there's inflammation and activation of what's referred to as the NF-kappa B pathway, which is important in inflammation. There's then satellite cell activation and muscle fiber regeneration, but eventually the regeneration of the fibers really can't keep up with the ongoing degeneration of fibers, and the fiber ends up dying and is replaced by fat and connective tissue, so also referred to as fibrosis or scar formation. Here on the right, you can see where these satellite cells are located just on the outside of the muscle cell. These satellite cells in the setting of segmental injury will increase in number, and they'll actually migrate to areas adhese to the muscle cell and actually form regenerating muscle fibers. And these muscle fibers, or these myotubes, will then fuse together and then reconstitute a normal functioning muscle fiber. But a regenerating fiber will have these nuclei that are more centrally located. And so that's one of the hallmarks when we look at tissue under the microscope, is we see these muscle fibers with central nuclei, which indicate ongoing regeneration. So I think a number of companies that are in the Duchenne space that are focused on dystrophin or microdystrophin restoration are really taking a 360-degree approach to the Duchenne patient and looking at biomarkers such as actual dystrophin, near full-length dystrophin production, or microdystrophin expression in the case of gene therapies, performance testing, which is really important in terms of the evaluating the functional benefits of therapeutics, non-invasive biomarkers, which can be blood biomarkers or other fluids, such as urine, can be evaluated as potential non-invasive biomarkers. Also, we have emerging imaging biomarkers such as skeletal muscle MRI, cardiac muscle MRI, and other imaging modalities. And then more recently, there's been activity monitoring, which has been employed with wearable devices such as the ActiMyo, which gives community-based measures of activity and function. One example would be the 95th centile stride velocity, which is determined by the ActiMyo device. This has actually been qualified as a clinical endpoint by the European Medicines Association, and there's attempts now at doing a similar qualification for the US FDA. And ultimately, we hope that we can measure the impact of these therapies on quality of life or patient-reported functional health. So these are assessments referred to as patient-reported outcome measures. So in terms of biomarkers, biomarkers are really important to drug development. And these are really tools that we use to give us an early indication that a therapy is perhaps having a benefit in muscular dystrophy. And so these are, again, indicators of normal biological processes. They can be indicators of pathogenic or abnormal disease processes, and they can also be indicators of biologic responses to a therapeutic intervention. And so there are a number of types of biomarkers. There are diagnostic biomarkers, which can give us clues to a specific diagnosis. There are prognostic biomarkers, which are measured at baseline, which give us risk for occurrence of the disease or perhaps disease progression. They can give us clues into what trajectory a patient may be on in terms of natural history. There are predictive biomarkers, which give us at baseline some indication as to the likelihood of a patient responding to a given therapy. There are also what are referred to as pharmacodynamic biomarkers, which reflect a biologic response directly due to a therapy or a treatment that's being investigated. And these pharmacodynamic biomarkers can be important in terms of determining the optimal dose of medications or biologic approaches. And then finally, surrogate biomarkers, which correlate with future clinical outcome. One example of a surrogate biomarker is dystrophin production, which has been deemed by the FDA to be reasonably likely to predict clinical benefit for a Duchenne patient. And there's actually 4 of the 5 approved therapies that are actually gene-targeted therapies actually have been approved based on the use of dystrophin production or a surrogate biomarker. The other therapy, Emflaza or deflazacort, is specifically approved for Duchenne muscular dystrophy as a indication. This was approved based on clinical and safety data from clinical trials. So there are a number of biofluids that can be used for biomarker analysis in muscle diseases. These could be fluids such as blood, most commonly, or urine, but other fluids could be evaluated as well. In the case of a muscular dystrophy patient, there may be, here's a muscle biopsy specimen on the left-hand side. There's active fibrosis. There's areas in the specimen that actually look fairly normal, but other areas where there's ongoing chronic inflammation. There's areas of oxidative stress and also areas of active necrosis and degradation of muscle fibers. And so these different biomarkers such as MMP9, microRNAs, CA3 titin, and so forth, myostatin, are actually indicators of these ongoing biologic pathologic processes that are in the muscle tissue. So to look just beyond dystrophin, there's these downstream effects of loss of dystrophin, such as reduced nNOS signaling, impaired mitochondrial function due in large part to calcium toxicity, calcium dysregulation, which can alter satellite cell activity, membrane stability, but also chronic inflammation and fibrosis. And many of the biomarkers are actually targeted at measuring chronic inflammation, fibrosis, calcium dysregulation, and impaired mitochondrial function. So these are downstream effects of the ultimate cause of the disease, which is the dystrophin deficiency. But with use of biomarkers, we can actually measure these downstream pathological processes. So just there are literally hundreds of biomarkers that are currently under investigation in Duchenne muscular dystrophy. I'm just going to give you some illustrative examples. Here on the left, you can see this is data we had published with Yetrip Halfett showing CK, which tends to decrease over time. As you lose muscle fibers, there's fewer fibers spilling CK, and the CK values will actually become diminished over time, as opposed to a healthy, typically developing control shown in the blue. MicroRNAs, for instance, microRNA 1, 133, and 206 have been shown to be elevated in Duchenne muscular dystrophy patients as opposed to control patients. And these microRNAs can actually be knocked down by dystrophin restoration approaches or by anti-inflammatory approaches. Urinary titin levels have been an overall indicator of muscle health. Here you can see the elevated levels of urinary titin versus a control population. So the idea here is that with a pharmacodynamic biomarker, there would be a predictive value in terms of the actual response to a dystrophin restoration therapy. And we could actually measure a biomarker that would correlate to actual increases in dystrophin production that occur over time. So there may be some biomarkers, such as biomarker 1, that actually decreases with dystrophin restoration. This, for instance, a biomarker associated with active inflammation may actually go down. There may be other biomarkers that actually may increase. So perhaps biomarkers associated with active muscle regeneration may actually show increases over time. And then if we actually look at animal models, we can actually perhaps use these same two biomarkers over a very much shorter period of time to give us idea of whether or not over a much less frequent period of time or a shorter duration period of time, whether the given therapy is having a positive effect on biomarker 1 or a positive effect on biomarker 2. So we can use these biomarkers in preclinical models where perhaps we can't directly assess some of the clinical benefits like we would in a human patient, but we can use biomarkers to give us these early signals for drug efficacy. Here's an example of a biomarker in association with gene therapy, the solid Biosciences gene therapy program. Here the therapeutic actually results in a dose-dependent increase in excuse me. Excuse me. So the here with the active gene therapy approach, we can see that the treatment actually results in a dose-dependent increase in dystrophin quantification or microdystrophin quantification and a decrease in plasma titin biomarkers with the gene therapy approach. So again, a biomarker example in a preclinical model in the MDX mouse model of positive response to a gene therapy treatment. Here's another example in a dog model of muscular dystrophy, again with gene therapy treatment. In this case, the similar gene therapy that's being developed by Solid Biosciences, a dose-dependent improvement in dystrophin production. Here you can actually do some functional measurements in the dog, but again, plasma titin levels, also with higher doses of the SGT-001, the gene therapy product, actually resulting in greater decreases in the titin level. We know that dystrophin levels or actual dystrophin protein is actually quite a bit shortened in Becker muscular dystrophy patients. Here is an example of a shortened in-frame transcript, so essentially a truncated or shortened dystrophin produced by an in-frame mutation. And again, these patients with Becker muscular dystrophy would have a much slower disease progression over time. The creatine to creatinine ratio has also been looked at as a potential biomarker. Creatine levels are elevated in Duchenne. The creatinine levels are decreased in Duchenne. So you can actually take a ratio of the creatine to creatinine ratios, and these can have been demonstrated to be abnormal in Duchenne, but also there are differences in Duchenne patients versus Becker patients. So here's an example again of creatine to creatinine ratios in a control population versus a Duchenne population. And you can see that in a variety of neuromuscular diseases, these creatine to creatinine ratios may be abnormal, but markedly abnormal in data on the right in the Duchenne population. And again, if you look at this creatine to creatinine ratio, there's really quite significant differences that have been shown to be seen or measured between Becker and Duchenne muscular dystrophy patients. Again, here, this really demonstrates that perhaps you could use this biomarker with a dystrophin restoration strategy that is trying to get a Duchenne patient to actually have a microdystrophin product or a shortened dystrophin product that would result in stabilization of the disease or slowing of the progression of the disease, much as what we would expect in Becker muscular dystrophy. And so biomarkers that perhaps can differentiate between Duchenne and Becker muscular dystrophy could be used to help us determine if a gene therapy approach is actually helping a Duchenne patient. And then finally, these biomarkers can actually be shown to be highly correlated with some of our clinical outcome measures, such as 6-minute walk distance, time function test, and so forth. And this shows the relationship between the creatine to creatinine ratio and 6-minute walk distance in Duchenne patients. So just to summarize with regard to biomarkers, these non-invasive biomarkers are detectable. They can be quantitated in Duchenne patients. We can use them in animal models in comparison to healthy control animals. There's a number of examples here. There's literally hundreds of biomarkers that are being evaluated. They change with patient age, disease progression, amount of muscle tissue regeneration. The candidate biomarkers have been identified using different technologies, different laboratories. But it's important to continue to discover these in the context of specific therapeutic strategies and validate these biomarkers. Again, this gives us a really important early signal with regard to whether or not a therapy may be helping a patient without having to perhaps conduct a 1, 2, or 3 year clinical trial. So we also were able to use these biomarkers as pharmacodynamic markers to actually tell us about optimal dosing of therapeutics. I'd like to just mention MR imaging as a biomarker. This is actually some fat fraction imaging in a Duchenne patient at 15 years of age. This is the upper arm in a 15-year-old, the same individual at age 16 and 17. And then down below, the calf muscle, the red tissue is the healthier tissue. The green tissue is the fibrotic or fatty tissue. So you can actually calculate a fat fraction in a leg muscle or in an arm muscle. You can actually see the active disease progression with regard to measurement of fat fraction in a Duchenne patient. And then MR spectroscopy techniques can actually quantitate specifically the fat fraction in a given region of interest in a muscle on an MRI scan. So here you can see the spike on the spectroscopy with disease progression showing an increased spike of the fat fraction in Duchenne patients. And just an example here, these are data from several patients, 4 patients that actually underwent MR imaging following gene therapy with the Sarepta 901 product. And you can see on the data images for each muscle with the data points on the left showing the fat fraction for control patients, the orange data points are the fat fraction for untreated Duchenne patients on steroids, and the blue data points show a normalization of the fat fraction in the patients that actually have been treated with gene therapy. So I think an exciting application here of a biomarker, an imaging biomarker, in the context of gene therapy. So I wanted now discuss new and emerging therapies in Duchenne muscular dystrophy. And in 2007, we really only had steroids available to us to effectively treat Duchenne muscular dystrophy. And since that time, there's been an explosion of therapeutic approaches for Duchenne muscular dystrophy. So dystrophin restoration strategies, which actually involve gene correction or gene therapy approaches, cell-based therapies. There's also been a focus on muscle cell fragility, enhancements of muscle bioenergetics or muscle metabolism, treatments which focus on inflammation and fibrosis. There are emerging specific cardiac treatments as well, medications which focus on calcium overload, and then with the RNA-based technologies such as exon skipping, a restoration of a normal reading frame and production of a near full-length dystrophin protein. So you can see really the explosion of therapeutic approaches to treating Duchenne muscular dystrophy. I think these can be broadly categorized as therapies which substitute or correct the primary dystrophin protein defect, such as exon skipping, nonsense suppression therapy, or stopcodon read-through therapy, gene therapy, and then we don't know whether utrophin upregulation will ultimately have an effect, but that has also been an approach to actually substitute a replacement or alternative protein for the dystrophin protein. And then a number of non-genetically targeted therapies which limit the progression of the dystrophic process, reduction in inflammation, prevention of muscle necrosis and fibrosis, increase in muscle regeneration, and improving mitochondrial health. And it's likely that we'll probably be looking at a cocktail of medications ultimately, which probably will include hopefully a dystrophin restoration strategy for a patient, depending on their genetic mutation, and then other therapies which focus on the downstream pathogenesis. So again, broadly, in terms of therapeutic targets, replacement of dystrophin, increasing muscle mass and regeneration, therapies which decrease inflammation and fibrosis, those that correct blood flow regulation, those that actually deal with calcium perturbation, mitochondrial dysfunction. So just to focus initially here on therapeutics which focus on inflammation and fibrosis, we know that prednisone and deflazacort are used really as standard of care. Inflaza has a specific Duchenne indication, but current trials have actually been conducted and recently completed with Vermourlon, which is a dissociative steroid, which hopefully has a improved side effect profile. We have data that hopefully will be emerging on the side effect profile of Vermourlon. Givinostat from Ital Pharmaco is an HDAC inhibitor, which enhances muscle regeneration, increases the size of muscle fibers, and modulates inflammation as well. Pamrevlumab from Fibrogen is a fully human monoclonal antibody that inhibits the activity of connective tissue growth factor. And then other therapeutics such as CAP-1002, which also have a positive effect on inflammation and fibrosis. So first of all, with regard to new therapeutics that reduce inflammation, Vermourlon, which has been developed by ReveraGen hopefully will be brought to market soon by Therapeutics as a dissociative steroid. There was a phase 2b study recently completed in 4 to 7-year-olds, 120 patients. The Vermourlon itself, like corticosteroids, inhibits inflammation and specifically the NF-kappa B activity. So it retains the efficacy due to the anti-inflammatory effect, reducing muscle inflammation. But hopefully with the chemical modifications, there is some improvement in the steroid-associated side effect profiles, such as bone fragility, fracture risk, growth stunting, weight gain, and so forth. So we still are awaiting some of the longer-term data from the Vermourlon trials, just looking at side effect profiles of Vermourlon. But initially, this data has been publicly disseminated by Reveragen and Santhera. And this actually shows a velocity in terms of the stand from supine activity. So a higher level is actually improved or faster rise time from the floor. And you can see here that this was the primary outcome measure in the clinical trial. The trial hit its primary endpoint. The 6 milligram per kilogram dose showed improvement over placebo, shown in the black, and also the 2 milligram per kilo dose, the lower dose, also showed significant improvements over placebo. And in fact, this is just looking at the absolute rise time in seconds, so sort of looking at it in a different statistical manner, but really looking at the absolute. A lower value here is actually improved function. And here you can see the Vermourlon treated patients actually had improvements in their standing times, which was very similar to that seen with prednisone there in the black dotted or dashed data points. So retaining the clinical benefit as seen with prednisone, but hopefully a medication with better side effect profile. And again, here is the data showing improvement in 6-minute walk distance with the Vermourlon treated patients versus the placebo treated patients. There are a number of other medications which are antifibrotics. They have a positive impact on muscle fiber growth. They can protect muscle membranes and stabilize the membranes, and also can act to have immune modulation. So one example would be Givinostat, a Tau Pharmacos medication, which is an HDAC inhibitor, which is being evaluated in phase 3 studies. CAP 1002, which will soon be evaluated in a phase 3 trial, a cell-based therapy, and pamrevlumab, which is also, that's a CTGF monoclonal antibody, which actually is in a phase 3 trial as well. So here, Givinostat, you can see the mechanism in the upper left. That is of a HDAC inhibitor, decreases activation of immune cells. It actually reduces fibroblast activation, restores regulation of cytokines and chemokines, and also has a positive impact on increased protein translation and improvement in terms of muscle regeneration factors. And in fact, the muscle biopsies have actually shown improvement in terms of fibrosis, necrosis, and also fatty replacement. The trial has been fully enrolled. This is some of the phase 2 long-term extension data showing a significant prolongation and ability to walk after more than 6 years of Givinostat. So this is the data on the left, our synergy data we published in 2018, showing a mean age with steroid treatment of loss of ambulation of about 13.4 years. And here the patients treated with Givinostat, median age of loss of ambulation of about 15.2 years on the long-term extension data. This is the phase 3 trial. I think very exciting data will be coming out in the summer of next year. And again, the trial was actually able to be truncated in terms of the sample size needed to be enrolled because of some of the preliminary data analyses surrounding the forced stair climb. So again, this trial is focused on 6 years, patients with Duchenne, 6 years of age and above, with given stair climb. There's a variety of functional measures such as 4-stair climb, North Star strength measures, time to rise, 6-minute walk distance, and also MRI as a biomarker as well, and close evaluation of cardiac function. And so due to the reduced variability seen in the 4-stair climb measure, the sample size was able to be decreased, and recruitment has been completed. The first data is anticipated to be available in the second quarter of 2022. There's another novel approach which will soon be in the clinic, which is being developed by Edgewise Therapeutics, which involves inhibition of fast myosin ATPase and fast fiber force development. So this is a therapy that actually, since most of the degeneration in Duchenne muscular dystrophy occurs initially in the fast twitch fibers, the fast glycolytic fibers, this actually specifically inhibits the force development in those fast twitch fibers. It's a fast myosin ATPase inhibitor. So that would essentially protect those fibers from more active degeneration. So again, these type 2 fibers, these fast glycolytic fibers are particularly susceptible to muscle fiber injury. The small molecule, in this case, the EDG 5506, or the Edgewise compound, selectively limits injury to those fast fibers by partially inhibiting fast myosin ATPase and force development. And in a variety of animal models, this medication has been shown to reduce muscle injury and prevent the accumulation of fat and fibrosis without really inhibiting physical function to a significant degree. So exciting there. There are also a number of therapeutics which are focused on targeting mitochondrial health in Duchenne dystrophy. This would presumably enhance the bioenergetics of the muscle fibers and enhance regeneration. Astellas is evaluating what's referred to as a PPAR delta agonist. Aperium Bio has active clinical trials in Becker muscular dystrophy, focusing on plus epicatechin. This is largely plant-based, and the company has been focusing on the development of plus epicatechin, which is a more potent form of epicatechin in relation to that which is typically derived from plants, or minus epicatechin. And then Biofluids as well has a therapeutic which has been targeting energetics. And then also CAP-1002, which is cardiosphere-derived cells, also has been shown to have positive effects on mitochondria as well. So two active clinical trials. Astellas has an active trial in Duchenne, a PPAR delta agonist, and plus epicatechin. There's been a trial conducted in Duchenne patients with positive effect on biomarkers, and then an active trial that has enrolled 23 patients with Becker muscular dystrophy and more mild forms of Duchenne muscular dystrophy over the age of 16 years of age. I'd like to finish by focusing on these very exciting dystrophin restoration strategies. Stopcodon read-through, which is the Adalurin medication, has been developed by PTC Therapeutics, approved in Europe. There are now antibody oligonucleotide conjugates with antisense oligonucleotides, PMO medications, which are actually approved. 4 of them are actually approved now by the FDA based on accelerated approval pathways. PPMOs, which are a next generation form of exon skipping, and then AAV microdystrophin therapy. So here's the stopcodon read-through compound, Adalurin. A number of clinical trials have been conducted in this. One ongoing trial, PTC 041, which has enrolled 250 patients. This is conditionally approved in Europe, for ambulatory patients with Duchenne. The FDA is open, I think, to approving this after reviewing future clinical trial results. I want to turn now to exon skipping, which is really the mechanism of 4 of the approved therapeutics in Duchenne dystrophy. So here you have a situation with normal dystrophin messenger RNA and normal translation by the ribosome of a normal full-length protein. So the ribosome assembling the protein. In the case of a deletion of exon 48 to 50, that causes an out-of-frame mutation. The ribosome comes to the impairment in the open reading frame and stops translating the protein. So you have a shortened non-functional protein which lacks its terminal binding site. And that protein then is unstable, essentially disintegrates, and you have complete absence of dystrophin because of disruption of the messenger RNA reading frame. So in the case, if you can pharmacologically essentially cover up exon 51 or skip it, you could actually bring the messenger RNA back into normal reading, produce a functional but shortened dystrophin protein, and that's really the idea behind exon skipping. This example being exon 51 skipping. So exon skipping in late stage clinical development programs actually address up to 30% of all Duchenne patients. Eteplirsen or Exondys 51 is FDA approved for skipping of exon 51. Golodirsen and Vitilarsen, both FDA approved for skipping of exon 53. And then casimersen, which Nicole mentioned earlier, Amondys 45, has recently been FDA approved PMO for skipping of exon 45. So these therapeutics actually address up to 30% of all Duchenne patients. And there are a number of companies that are currently in this, using this strategy of exon skipping with mutation-specific interventions. Sarepta has the approved therapies as well as what is referred to as a peptide conjugated PMO that's in clinical development. This is a next generation therapeutic, PTC with its adalurin, a stopcodon read-through compound. NS Pharma has a couple of, they have an approved agent that treats exon 53, and then others that are in their pipeline, for instance, an exon 44 skipper and so forth. There's also Avidity Biosciences has new technology, an antibody oligonucleotide conjugate for enhanced tissue selectivity with animal data actually showing significantly increased levels of dystrophin production with perhaps less frequent dosing. And then Trotta Therapeutics has their endosomal escape vehicle technology, which actually many times the antisense oligonucleotide medications are trapped in these endosomes and degraded, and they never really get to the nucleus to actually try to work to produce dystrophin. And Trotta Therapeutics has this EEV technology or endosomal escape vehicle technology. So currently, again, active clinical trials that are underway in Japan, an exon 45 skipping agent. Sarepta has their PPMO trial. And then Avidity Biosciences and Trotta Therapeutics are both poised to enter the clinic initially with exon 44 skipping agents. But I think it's really very exciting. We have these next generation exon skipping compounds, PPMOs, which actually are peptide conjugated morpholinos, which actually drive more of the PMO into the cell, into the muscle cell. These antibody oligonucleotide conjugates for tissue selectivity that Avidity Biosciences is developing, and then the endosomal escape vehicle technology being developed by Trotta Therapeutics. Here's an example of the PMO or the peptide conjugated platform where a cell-penetrating peptide is actually attached to the PMO. So this is the so-called PPMO, and there are active clinical trials looking at a exon 51 skipping PPMO medication. And then to conclude, I'd like to just comment on the AAV gene therapy approaches. There's at least 4 companies with Sarepta, Pfizer, Solid, Adentis, and Genethon actively involved in this space. These are very exciting therapeutics under development. The essential components of gene therapy involve a bus to transfer the gene, the replacement gene. So you have to essentially have a vector or a viral vector to deliver the transgene or the replacement gene to the target tissue, a tissue-specific promoter that drives expression into the intended tissues, and then the actual replacement gene or the transgene, which must actually replace the critical functioning aspects of the dystrophin protein in this case. So essentially with AAV, this would be the full-length dystrophin, the full-length copy DNA, which is a huge gene. The AAV capacity, on the other hand, has really limited carrying capacity. It can't carry the full-length copy of this dystrophin protein. So this would be an example of a slightly shortened dystrophin protein, but again with the essential binding domains, the amino terminus on the left, which binds onto actin, and the carboxy terminus on the right, which binds onto the series of dystrophin associated proteins. And in Becker dystrophy, these patients again have limited disease severity. So this would be the example of a small dystrophin or a microdystrophin, which was actually observed in a 61-year-old ambulant Becker patient. Had an incredibly shortened dystrophin protein, but had the critical binding regions and critical hinge regions. The actin binding domain was still present in this patient. This patient had hinge regions, a sarcolemmal binding domain, and other hinge regions. And this patient actually continued ambulating. So this is really the replacement gene, which is being constructed by companies in the gene therapy space. And there are variations of this gene therapy for the purposes of clinical trial development. If we go here to the next, just to show, here is the full-length dystrophin protein showing the protein domains of dystrophin. These are the exons that are associated with the protein domains. You can see here exons 1 through 8 are involved in the actin binding domain of dystrophin, and there's other hinge regions and sarcolemmal binding domains, as well as NOS regions, and then a carboxy terminus domain. So again, if you look at the full-length dystrophin protein, the full-length protein really cannot be packaged into an AAV carrying vehicle. Here you can see the example. Here, Mickey is actually going on a vacation and can't fit everything he wants to take along in the suitcase. So the example on the left here shows the full-length dystrophin with its specific regions of importance: the hinge regions, the actin binding domains, the sarcolemmal binding domains, the NOS binding regions, regions associated with microtubule organization, and a carboxy binding domain. This was the small dystrophin or the microdystrophin seen in a patient who was 61 years of age with mild Becker muscular dystrophy. This patient had a deletion for exon 17 through 59. And then just an example of one of the microdystrophin constructs here, in this case, the Sarepta construct, which has been sort of patterned after this construct developed for treatment. So again, the microdystrophin has these essential regions which actually can fit into an AAV. There are 3 different transgenes which are currently under investigation and development by Sarepta, Solid Biosciences, and Pfizer. Sarepta actually uses the RH74 viral transmission vector, and Solid and Pfizer both use the AAV9 viral transmission vector. And here is an example of the Solid Biosciences microdystrophin construct, which actually maintains the nNOS binding domain in their microdystrophin construct. So all these have been developed with some rationale in mind, and all have preclinical data demonstrating effectiveness. Here's just an example in some of the first patients treated with the gene therapy construct by Sarepta. This precipitous drop in CK value indicative of stabilization of the sarcolemmal membrane. So this is, again, use of a biomarker to demonstrate biologic activity. And this would be another more biomarker data, which has been publicly presented by Sarepta, showing essentially vector genome copies per nucleus, the percent of normal expression by Western blot, the percent of dystrophin positive fibers, and the percent of dystrophin intensity by immunofluorescent staining. So just another example of biomarkers used to demonstrate efficacy. We need to be aware of potential safety issues with the AAV gene therapy programs, such as nausea, vomiting, thrombocytopenia, liver inflammation, and injury, complement activation, along with potential risks of hemolytic uremic syndrome. And then I think also we need to be aware of potential immunologic response to the transgene itself. And some of the programs have actually in the past excluded patients, for instance, with deletions of exon 1 through 17 and so forth, because of this immunologic risk of risk of a immunologic response to the transgene product. I think in terms of key issues with microdystrophin, I think, again, safety is being evaluated. It's still largely unknown. We know that there is some of the programs that have had issues of complement activation, liver toxicity, coagulopathy. The efficacy of these highly internally deleted dystrophin proteins, the microdystrophins, have not been fully established. Also, we don't really know the durability of the effect at, say, 2 years or 5 years. That's still somewhat unknown. There may be decreased expression that could occur with somatic growth of muscle fibers. There's antibody titers or previous exposure to AAVs, such as the RH74 AAV or AAV9, which could put a patient at risk, and that thus this impacts the eligibility. The immunization or treatment with this one-time dose could likely preclude re-administration of the dose unless we take other approaches such as plasmapheresis or treatment with immune modulating therapies to allow redosing. I think that's going to be a active focus of investigations in the future. I think dosing extrapolation to older patients is largely unknown. A lot of these gene therapy treatments are dosed on a per kilogram basis, but if a lot of the body weight consists of fat tissue or fibrotic tissue, we don't really know how the dosing extrapolates from, say, a 4-year-old to a 15 or 16-year-old patient. And then also there's a question of, does the AAV gene therapy actually transduce the satellite cells, which are active in regeneration of muscle fibers? And then again, we should be aware of the different quantification approaches of Western blot versus mass spec. And then just to conclude, I'd just like to focus on a therapeutic that has targeted the non-ambulatory population, or the later stage patients that have some impaired degree of overhead reach. The goal of this therapy is to maintain ability of upper limb functions, such as hand-to-mouth function and distal upper limb function, prevent the progression to 24-hour-a-day non-invasive ventilation requirement. Most patients are I think, appropriately treated with non-invasive ventilation. So this therapeutic area really is focused on these early non-ambulatory patients and late non-ambulatory patients. I just want to point out, based on the Parent Project Muscular Dystrophy patient and caregiver survey, the ability to bring the hands to the mouth were noted by the patients themselves to be the second most important issue to quality of life. Repositioning oneself at night was the most important. For the parents of the non-ambulatory patients, the hand-to-mouth function was the most important, and then repositioning the body at nighttime was the second most important. So again, a lot of the non-ambulatory trials are using the performance of upper limb measure, and this demonstrates the entry criteria based on the performance of upper limb measure, the so-called PUL, and these therapies are largely targeting patients that have some degree of upper limb impairment at baseline, so you can demonstrate improvement with the therapy. And the therapies are also largely targeting patients with retained hand-to-mouth function. So PUL entry score of 5, which is hands to scalp, all the way down to a PUL entry score of 2, which is getting an unweighted hand to the mouth. The primary outcome measure has been the performance of upper limb. There's a new version which has been shown to have better measurement qualities than the 1.2 version, the PUL 2.0 version. And then I just want to end by talking about CAP-1002, which is a stem cell or a biologic, which doesn't work by engraftment or engraftment of the stem cells, but actually these cardiosphere-derived cells actually are infused intravenously. The cells themselves go to the pulmonary vasculature, where they then secrete what are called exosomes, which are these membrane vesicles that contain microRNAs. They also obtain a variety of growth factors and factors that can actually drive the efficacy more systemically with both cardiac tissue as well as skeletal muscle tissue. So the CAP-1002 targets myocyte loss, nitrosyl active stress, inflammation, mitochondrial inefficiency, microvascular ischemia, and fibrosis. And we conducted the so-called HOPE-2 clinical trial, which enrolled 20 patients. They were randomized to receiving the active intravenous cell-based product versus placebo. These patients all, if they were ambulatory, they had 10-meter walk times that were greater than 10 seconds. So they were late ambulatory. Their PUL entry scores were 5 down to a PUL entry of 2. And these patients were all on steroids for 6 months. And here is the effect. The blue data points are those treated with CAP-1002. The orange data points, those treated with placebo. And this demonstrated the data statistically significant improvement in preservation of upper limb function with a p-value statistically of 0.01, 71% slowing of upper limb disease progression. These were the variety of the PUL measures. To the right is favoring the CAP-1002, to the left would be favoring placebo, when virtually all of these PUL outcome measures actually favored statistically the CAP-1002 treated patients. And this was actually the data on using cardiac MRI with essentially stabilization of the left ventricular ejection fraction by cardiac MRI, essentially 107% slowing versus natural history. So you would expect very modest decline, nonetheless, of say 3 to 4% in left ventricular ejection fraction. But this data actually demonstrated stabilization of heart function with the cell-based therapy. And these were a variety of biomarker, imaging biomarker evaluation using cardiac MRI. Again, the vast majority of them favoring CAP-1002. And in fact, the creatine kinase MB to total creatine kinase fraction, or the MB fraction, actually also was a biomarker that favored CAP-1002. So with that, I will end, and I think we have a few minutes to take a few questions. Thank you very much.
Thank you, Dr. McDonald. That was awesome. I loved all that data. It's amazing how busy the field is right now surrounding all of Duchenne. We do have a couple questions from our audience, and let me just start with the first one that I received here. This person says, I've not heard before the impact of the muscle cell breakdown causing a massive influx of extracellular calcium. Is this one of the reasons why osteopenia and osteoporosis is an underlying issue with Duchenne?
Yeah, it's a good question, but really the influx of calcium really pertains more to the muscle fibers themselves, and this cascade of events that occur when you have tears in the muscle membrane. I think the issue of calcium related to bone health is really a separate issue that's really due to probably a combination of the paralysis of muscles, the lack of weight bearing through bony structures, and then also the standard of care treatment with steroid medication. But that's a good question, but I think we need to differentiate the calcium issues on the muscle fiber versus those that pertain to bone health.
Okay. Is there any data that tells us that there is a better age range to infuse Duchenne boys with microdystrophin?
Well, I think there's emerging data that probably suggests that the earlier we can treat patients, the more effective these treatments will be. I think the clinical trials initially have focused on 4 to 7-year-olds, but we're hopeful that use of these dystrophin restoration strategies with gene therapy will actually help stabilize the disease for later ambulatory patients as well as non-ambulatory patients as well. I mean, my sense is that if we treat boys younger, we can actually see improvement in function versus what we'd expect in normal disease progression and natural history. If we treat boys later, I think what we'll see is probably stabilization of function and hopefully prevention of functional decline in these patients.
Okay, and I think that might roll into this comment that we got from Gordon in our audience. Is there any new therapies for older adults with Duchenne over 30? That's
a great question. I think that it really probably depends on, first of all, I think some of the cardiac therapies I think would be very appropriate, for patients who are in their adult years who still have progressive cardiomyopathy, and also preservation of distal hand function is extremely important, I think, to men with Duchenne muscular dystrophy. Just an example of that is that in looking at our long-term steroid data, if patients continued steroids on into adulthood, their median age at losing distal hand function was 31 years of age, versus those who were not treated with steroids who lost distal hand function at 23 years of age. There was about an 8-year prolongation of distal hand function. So I think a lot of these therapeutics potentially could serve to preserve distal hand function in men with Duchenne.
Very interesting. Okay. Are there any gene therapies being developed for exon duplication mutations?
I think generally, I think the feeling is the AAV gene therapy approaches are applicable to duplication mutations as well as deletion mutations. So it's really potentially applicable to both patients with duplications as well as patients who have deletions. I think initially what we'll probably see is some subpopulations possibly initially not treated with gene therapy just because of risks of immune treatments. But I think the companies are going to, in the very near future, probably target those populations with some new immunomodulatory pharmacologic strategies used, so some medications used in addition to steroids to help prevent the patient from having T-cell mediated immunologic reactions to the transgene product. The point is, I think duplication mutations have been targeted as well as deletion mutations in the AAV gene therapy programs.
Okay. Do you happen to know what the status of Solid's transgene being used in partnership with Ultragenyx with a different vector? Do you have any information on that?
I believe I had just heard that they were working on a strategy using an AAV8 vector as an alternative to the AAV9 vector, with, I think, using the same transgene, which has the nNOS domain that's is still present in the transgene. That's what I know about that to date. That's a good question.
Okay. The upper limbs report that you did with the CAP 1002, is that available anywhere for families to locate, or is that still?
We have presented it publicly. I'll have to talk to Capricorn. We have submitted this for publication. It's currently under review by a peer-reviewed medical journal. We hope to get this published in the very near future because it's very important data.
It is. It's very important. We have a question. Does gene therapy have any implications for cardiomyopathy and bone density issues?
I think certainly the gene therapy there is likely going to be, some production of the microdystrophin in the heart. That I think the trials have actively obtained measurements of cardiac function. But I think there's also some active gene therapy approaches that are specifically targeting the myocardium of the heart that will really be, specific gene therapy strategies to try to get effective dystrophin replacement into the myocardium.
Okay. Let me see. I'm just kind of going through the chat here real quick. Could you explain endosomal escape therapy?
Yeah, I think with a lot of the, traditionally, the thought is that with many of these therapeutics, not just exon skipping, but with a lot of medications, the molecules are internalized into the cell by it's taken up in an endosome, which is a vesicle that sort of takes the molecule up, and the molecule actually has to get out of the vesicle in order to sort of make its way to the nucleus where it would have its effect on exon skipping. And the idea is that a lot of, the thought is that a lot of exon skipping strategies or drugs, actually, the exon skipping drugs, they make it into the cell, but they never make it out of the exosome. So the endosomal escape vehicle technology is a methodology to actually try to effectively get the exon skipping drug out of the exosome, so greater proportions of the exon skipping drug can make its way to the nucleus, and perhaps patients can be dosed less frequently, perhaps once a month or even less frequently, and then also perhaps this approach would lead to greater dystrophin production and greater efficacy. So that's the idea there.
Okay, thank you for explaining that. Could you, so I think this is our last question here. Can you explain why transgene immune response is only a concern with certain deletions? I think
this was data that was originally presented by Dr. Jerry Mendel back in 2010 with and he found that there were some patients with early deletions between 1 and 17, which actually there was a couple of patients that actually showed evidence of some immunologic response to the transgene. So that was really the rationale for in Sarepta's study 102, while they actually only included patients that had deletions inclusive of exon 18 to 58, because the idea would be that essentially there would be some degree of there would not be any previous exposure to those epitopes of the gene, and so that would reduce the risk of an immunologic reaction to the transgene product. So that was sort of the concept behind that. I think the companies have all differed in terms of their approaches to specific genetic exclusion criteria for their clinical trials. We'll await how the companies are going to approach this moving forward. But it seems that at least some of the case reports, and particularly the study done by Dr. Mendel has sort of focused on some of those larger deletions in the earlier part of the gene as being producing a potential risk of immunologic response to the transgene. So that really was behind, I think, Sarepta's exclusion criteria of exons 1 through 17 and 59 to 79 in their 102 clinical trial.
Perfect. All right. Well,
and again, that isn't to say that those patients with those mutations could never get gene therapy. It just means that perhaps a company might come up with a different immunologic or immunosuppressive strategy to approach that population beyond just treating with high-dose steroids.
Sure. Okay. Well, thank you so much. It sounds like there's a lot of hope in the future coming down the pipeline. There's a lot of things being investigated and being studied. So, and I think it's great. You sound very hopeful about the future for these Duchenne kids.
Oh, absolutely. I've been treating these patients for 35 years, and it's really unbelievable. I think the hope that we all have at this point in time is really amazing.
Well, thank you so much for your time. I know it's early where you're at, so I definitely appreciate you hopping on this morning.